Hello everyone, good morning to all. Guys, please clear me. Audio video clear ଅଛି ନା? Pranam guys. Khushbu, please tell me audio and video is clear na? Yes ma'am. Okay. So we'll start the class. So today's topic is guys genetics. Okay. So let's see what is genetics. So before starting the MCQs round, so I have some theories about it. Like बहुत ही कम theory जोटा कि तम मानन को टिके boost करिवो तार MCQs round cover up करिवा को clear करिया ପାଇଁ. So not delaying, start the class. Guys, see what is genetics is actually. Genetics is the branch of biology. We all know that is either we are relating with the plants or with the animals. So genetics is the branch of biology that studies heredity and the variation of trait in organisms. Any kind of organisms that can be. And it focuses on how genes, the units of inheritance, the units of inheritance are passed from parents to offspring. That means from our ancestors to next offspring zone ko amara transfer hochi the genes, the hereditary character. Suppose you are tall as your father is tall. Suppose your hair is curly as your mother's hair is curly. And you have a very sweet and clear blue eyes as your grandparents or the grandmother or grandfather have the same eyes. So this is all because of what? The genetical hereditary characteristic transformation due to the ancestors to the offspring. So influencing traits and biological process, this is all about the genes and the genetics. Next, see what is actually the gene start kotu heisila. So guys, gene, a segment of DNA, that is a segment of DNA that contains instructions for building and maintaining an organism. Suppose मैंने जानी थी कि suppose DNA रा structure को type रे रहुची. This is the double helical structure and suppose this is the set. When it is completely, this is not joined anywhere. The segment, both the strands are not joined anywhere, because if you will have the 3D animation structure of the DNA, you can clearly visible that. So there is no joint inside that, and the whole segment, the set of the segment, we can say that is the gene. So a segment of DNA that contains instructions for building and maintaining an organism, that is called guys what? Gene. And DNA, what is DNA guys? This is the deoxyribonucleic acid, the molecule that carries genetic information in cells. Like inside our body, in every kind of tissue we are having the cells. And the cells रे problem कौन हुई? ना DNA carry करे cells. The molecules that carries the genetic material, the genetic information in cells, that is the DNA. And it consists of sequence of nucleotides, the nitrogenous base, that is the ATCG. And obviously what is ATCG stands for, you will know in further. So that encode biological information. And in case of chromosomes, guys, this is the very crucial theory. You have to know that what is the chromosome, because exam रे हमारे MCQs एमिति आसी जाए कि pairs of chromosomes, or if you are saying the 23 pairs of chromosomes, then tell me the number of chromosomes. So simply 23 into 2, there is 46 of chromosomes in numbers, but in the case of pairs, this is only 23 pairs. So structures within cell that organize and carry genetic material. The humans have 23 pairs of chromosomes, and throughout the chromosomes, all the hereditary character are transmitted through. Okay. Next, what is the alleles, guys? What is the alleles? So different versions of a gene that determine specific trait. So different body, different figure have different alleles. They have the different characteristics, they have the different DNA segment, they have the different fingerprint also. So due to what? Due to the presence of alleles, all the segments can be differentiated easily. And guys, what is the genotype and the phenotype? The genotype refers to the genetic marker of an organism, while phenotype is the observable physical or biological or biochemical characteristics. Keep it in your mind, what is the genotype and the phenotype. यहाँ पर question आसीबा and we will answer the same. So genotype refers to the genetic, genotype ru genetic and phenotype ru physical or biochemical. Pheno ru physical, geno ru genetic. Clear? That's it. Next, see the Mendelian genetics. So genetics को first हमारा propose किए करते. The father of genetics is known as the Gregor Mendel. So first time the term genetics who was proposed by, that is Gregor Mendel. That's why he was known as the father of genetics. Established the fundamental principles of heredity through his work on pea plants. So first he researched on pea plants and succeeded that research and concluded in the genetics. There are two methods, that the law of segregation. Basically there are three methods, we can have a look in further. But first of all, there the law of segregation. What is law of segregation? Each organism carries two alleles. Each organism carries two alleles for a trait. For a single trait, there is a two organisms which are consist of different alleles, then consist of a single alleles. That is called the law of segregation. And we segregate during gamete formation. Further method, they will form the gamete. So ensuring each gamete carries only one allele. Keep it in your mind that law of segregation differentiate कौन करूची? Dita divide करूची, like each section have carry the one allele. And further divide है कि जितने gamete formation करूची and ensure करूची कि दीटा allele combine करिके गोटे single allele रे convert हेई परिबो. गोटे single gamete formation ने गोटे single allele रही परिबो. That is the law of segregation. That also we will read in this class. And the law of independent assortment. What is that's also a step of Gregor Mendel's genetic theory. Guys, the genes for different traits assort independently. Different traits पाई assort करिबो independently हमारा कौन है ना genes of another during gamete formation. Suppose gamete formation procedure रे जेतेबेले हमर single दीटा allele मिसी कि गोटे single allele बनुथिले दीटा different allele. But एठी कौन होची ना different trait पाई independently dita separate gamete formation करूची. ये ना independent assortment. So what's about the modern genetics and applications? Guys, the genetic engineering, the human genome project, the CRISPR-Cas9 technology. These are all about the modern genetics and the applications. That is also proposed by the Gregor Mendel, the advanced modern genetics we can say. So the genetic engineering, like basically in the UPSC or OPSC, the questions is very common for their perspective, that what is the genetic engineering, what is the human genome project, and the role of CRISPR-Cas9 technology. CRISPR-Cas9, guys, is a molecular seizure we can using for the editing or the gene silencing purpose. So genetic engineering is what? The deliberate modification of an organism's genetic information using the biotechnology, like any kind of modification. Suppose you have a very defective body or you have a defective gene structure. When you are going through the biotechnology, then it can be edited, it can be coated, it can be silenced, it can be suppressed. So this is all about the genetic engineering. And applications include agriculture. Agriculture, we can see the PT cotton, or the medicine example, we can say the gene therapy, and in the industry, golden rice, the fortified milk, everything we will see in the genetic engineering class. So these are the just brief idea about you, about the modern genetics, what is the modern genetics and what is the traditional genetics. So human genome project is what? An international scientific research project aimed at mapping and understanding all the genes of the human species. Like different, like human species बहुत ही vast. तो सेमानक पे research करिवा लगी we have a strong scientific research team. So that's the aim to mapping and understanding the all genes, like varieties of genes. Suppose gote nua trait asuchi tar genes komti different hoji than the older trait. So ei sabu par research karuchi kiye na, this is the HGP and the human genome project. And guys, what is the CRISPR-Cas9? What is CRISPR-Cas9? Guys, a revolutionary gene editing technology. As I have said it before, that it is nothing but used as a molecular scissor for gene editing, for gene silencing, for gene suppressing, etc. So that allows for precise, efficient, and cost-effective modification of DNA. Guys, if you have any kind of hereditary disease and you want to recover that after long periods of medications, you are not recovering from the first one, or this is so ancestral. So taku cover up kariba lagi, taku treatment kariba lagi, the process using that CRISPR-Cas9 technology and the used as a molecular scissor here CRISPR-Cas9. And suppose the gene is here and एठू तमे किछि portion cut करूछ and ए जो cutting पे use करूछे, that is the molecular scissor and CRISPR-Cas9. and again तम माने भाबुथो मैम cut down होला परे पुनी again सेतरे जो fix हबो केमिति हबो, there is a need of glue. so glue आकारे कोन यूज हुए ना, ligase. so ligase we can use as a glue after cutting by using the molecular scissor CRISPR-Cas9. okay, so this is all about what the brief idea about the genetics. Now we will come to the MCQs round. So we will discuss as of 40 questions. Okay, so starting from the first one, tell me in the comment section, guys, who is known as the father of genetics? What I have just told you. Tell me the right answer in the comment section. Who is known as? So tell me the right answer in the comment section, that who is the father of the genetics? Who is known as the father of the genetics? Tell me. Odisha, you are in the class. After a long time, I am seeing you. So, Sobhan, class re kehi thandu na thandu, Sobhan mo class ke miss karo na. So glad, huh? Okay. So guys, see, who is the father of the genetics? As I have just said, that Gregor Mendel is the father of genetics. So Gregor Mendel, considered the father of genetics, studied about what? The pea plants. On which plant Gregor Mendel researched and succeeded in the research of genetics. Guys, MTB question has said that is pea plants. Okay. And to learn about the transmission of traits from parents to offspring. For what purpose? He started his research to learn about the transmission of traits from parents to the offsprings. And the trait genetically determines the characteristics. Characteristic is what? A heritable feature about your hair color, about your hair texture, about your eye color, everything, about your height, weight, everything. Okay, this is about the genetics. So, to research, to know more about the genetics, he started research on the genetics. See, what is the basic unit of heredity? Tell me the right answer in the comment section. What is the basic unit of heredity? Is that chromosome? Is that gene? Is that DNA? Or that is nucleotide? Tell me, what is the basic unit of heredity? Yes, that is gene. What is the basic unit of heredity? The gene is the basic unit of heredity, the starting unit you can say. That is the gene. See, the unit of heredity that controls the development of one trait लेते बेले सिंगल लाइक तार गोटे गैमेट रो स्टार्ट हुए तार सेल ग्रोथ सेते बेले ताकू किए रिस्पांसिबल हे थाए तार जीन ग्रोथ पाई दैट इज सॉरी तार ग्रोथ पीरियड पाई that is gene that is made up of what DNA and the DNA made up of what the cell and the we can say sorry not cell the genomes okay the set of genes are called the genomes. Next see which of the following is not a nitrogenous base in DNA. Which of the following is not a nitrogenous base in DNA? Guys, I have told you there is a 4 type of nitrogenous base both in DNA and in RNA. So DNA re present rahibo AT, C and RNA re rahibo AT, UZ. Okay, so there is a just replacement of the uracil and the cytosine. No, uracil and the guanine. So what will be the guys? So which of the following is not a nitrogenous base of DNA? So कोटा नुआ adenine रहिबो, thymine रहिबो, cytosine रहिबो, even कौन रहिबो ना guanine भी रहिबो, but रहिबनी तो कोटा रहिबनी तो uracil because uracil is the nitrogenous base of RNA that can be replaceable instead of guanine in case of RNA. See, so there are 5 nitrogenous base in total. Actually रे 4 but DNA and RNA को mix करिकी हमारा कौन ना guanine, adenine, cytosine, thymine and uracil को add करिछि but in set of DNA there are 4 and in set of RNA also there are 4 types of nitrogenous base. That is the in DNA guys the guanine, adenine, cytosine and thymine. But in case of RNA there is guanine, adenine, cytosine and uracil. So not I have said you that guanine replacement karuchi uracil ko no guys the thymine replace karuchi uracil. So in DNA there is a T, C, G and RNA there is the A, U, C, G. Okay, so the guanine and adenine have the structure like the double ring structure and they are called as purines. एमितिरे भी question आसी जाई पारे कि what are the purines? Okay, you have to say the purines are the guanine and adenine present in the DNA only. And in case of pyrimidines, the single ring structure kouthi asibo in case of RNA. And the cytosine, thymine and the uracil are consisting together to form pyrimidines. Okay? Yes, sir. Right. Next question. In humans, how many pairs of chromosomes are present? In humans, how many pairs of chromosomes are present? I have just told you about the elaboration. Guys, kete rahiba? Na 23 pairs of chromosomes rahiba. But jodi calculate kariba how many chromosomes, then convert heijiba 23 into 2. There are 46 chromosomes. But pair kahile there is always 23 pairs of chromosomes. Okay. So humans have thousands of genes located on 23 pairs of chromosomes in our cells. Each cell in our body except sperm and egg have the same 23 pairs of chromosomes in their nuclei. Okay. Next, see guys. Which molecule carries amino acids to the ribosome during protein synthesis? Tell me which molecule carries amino acids to the ribosomes during protein synthesis? This is transfer RNA. Guys, carry out karibo. That means kono transfer karibo from one to another. That means ke karibo messenger ta karibani, ribosomal ta karibani, DNA not for a once. So ke karibo na transfer RNA obviously. So let's see. So tRNA work kon rahuchi na tune the precise codon reading, stabilize codon anticodon interactions, increase the capabilities of transfer RNA to decode multiple synonymous messenger RNA codons, allow transfer RNA to rapidly react environmental challenges, so etc. You can have a look here that these are the functions and the disorders can be formed due to the deficiency of the transfer RNA in our body. Okay, so before that there is a short notice guys about our plan guys. Just have a look guys. जो माने भी interested अछंती हमारा batch already launch हो जाइछि like आज ही last date जो माने इंटरेस्टेड अछंती बैच परचेज करिया पई सिंपली समानु जाई कि StudyIQ चूज करबा को पड़िबो. Then here you have to click the first option of StudyIQ. Then if you are the new, then you have to log in, else you have to sign up in the my account section. So here guys, okay, so here just search as OPSC. Guys, the Paripurna batch is live. The batch 3 is just live now, and today is the first admission date is ending till the last date was today. So quick and hurry up, just enroll the batch by using my code. You can have the least discounted price. Just click the buy now here, and you have to go to the offer availables, and here you have to click as SSP live and have to apply here. And you can see that the least discounted price, that is the 9,999, and you can use my code SSP live to avail the offer. Okay, and in the same way guys, in the OPSC section, guys, there is a sociology optional, that this is also 16,999, you will show up in the platform. But if you are using my code as after clicking the buy now, there are 3 offers available, but you have to put my code SSP live and have to choose apply. After that you can have a discounted price, that is 9,999. Okay, so this is all about the offers. And if you are interested to do our Aarambh batch, to take the purchase about the Aarambh batch, guys, see here is the Aarambh badge also. Okay, here you can enroll the batch by using my code, that is here you can choose SSP live and apply. Here also you can have a 9,999 is the least discounted price, and here are the top educators have taken your classes. And guys, if you are interested like the mock test, that is the Abhyas, you can also preparation को जदी strong करबा को चाहूछ, the mock test guys एमिति अछि हमर सिस्टम कि परिपूर्ण बैच नौच या फिर Aarambh बैच नौच, जदी तमरा प्री क्लियर हे जाउछे and mains रा preparation को तुमको टिके सपोर्ट भी मिलिबो and free रा whole amount तुमको refund मिलि जिवो. With that 23 sets of तमरा free mock test तमरा रहिबो. Instead of that तमने जदी preparation को आहुरी strong करिया को चाहूछ, आहुरी extra mock test देबा को चाहूछ, then you can purchase the batch, that is the Odisha PSC Abhyas batch. The code will be apply करो न करो तुमको code एतिकी 2,800 रे मिलि जिवो. Okay, so thank you for this, and let's start the class again. Guys, the next question, so what is the shape of the DNA molecule? Guys, DNA molecule have a always double helical structure. That means this is single helix, no double helix, triple helix, linear strand, this is double helical structure. So the double helix structure amara kon achi DNA re, the guanine, cytosine, thymine and the adenine. Okay, and the double structure, the strands and the double strand structure, the strands are contain the sugar phosphate backbone and the base pairs. Base pairs are so the 4 are the nitrogenous bases with the sugar phosphate backbone. Clear? Next, which process converts DNA into RNA? Which process converts DNA into RNA? That is the translation, that is transcription, that is the replication, that is the translocation. Which process converts DNA into RNA? This is transcription. Right, Soban. This is transcription. Okay. So transcription केते type ର रहुଛି, sorry, transcription कौन होଉଛି, either transcription method or translation method. So जो case टा हमरा कौन कहूची ना converts करूछु DNA to RNA, DNA into RNA, then RNA into DNA. These are the process of transcription. So DNA to RNA करूची कौन ना हमर जो nitrogenous base अछि, just से nitrogenous base is responsible for unzipping the DNA double helix during replication. So during replication, the unzipping of DNA, this enzyme is responsible for such kind of work. The DNA polymerase, the helicase, the ligase, or the primase. Tell me the comment section, the right answer. Yes. Yes, so one, this is helicase. So see, so a helicase is an enzyme that unwinds double-stranded nucleic acids, that is DNA to RNA re convert karibare taku helps kore into single strands. This process is crucial for various biological functions like DNA replication, repair, and the transcription. So both in like in DNA replication hela and DNA repair hela, DNA transcription hela, in those process we are using what? Helicase as an enzyme to unwind the double strand of DNA. And helicase use energy from the hydrolysis of nucleoside, the triphosphates, to separate the nucleic acid strands. Okay, this is all about what? The helicase. Next, see, what is the term for different forms of a gene? What is the term for different forms of a gene? Guys, that is alleles, loci, or the chromatids or the genotypes. Guys, the term we are using for different types of a gene is what? Is the alleles. Okay, see how? Guys, what is an allele? An allele is a variant of a gene controlling the same trait. Alleles usually occur in pairs depending on how they are expressed, that may be dominant or the recessive. That means suppose you are given an example as the tall plant and the dwarf plant. The T is the capital and the dwarf pine, the T is the small. So even you are showing the T for the same times, then this is the dominant one. Suppose in the cross section I am showing you this one. Not this one. So when the capital T is formed 3 times, then the T, that means the tall plant are the dominant here, and the small t you can see there is a 1 time. That means they are the suppressive, or you can say the recessive one in the case of alleles formation. So offsprings are alleles for the flower color, where R is the dominant and R is the recessive in the allele. Suppose for example ame jodi neba RR for red and RR, small rr for white and capital RR for red. Then in the case of the chances of getting the pink flower, so one red and white, consider the pink flower, the recessive and dominant, then we can consider like the T, then we can say the R and the RR, we can have the RR again with the RR, and guys here is the RR. So all we can find that the pink flower, because everything is compulsory, the capital and small, the pink and the white and the red are compulsory and equal. That means the chances of having pink flower is much more than the having not a pink flower or having any single flower of white or the red. Okay. Next question, see, which of the following is purine base. Which of the following is a purine base? Adenine, thymine, cytosine or uracil? So guys, purine and pyrimidine दीटा तुमको कही guys purine रे कौन रहुची ना instead of आमर purine रे adenine रहुची तो ए सब गुडा को हमें कौन कहि परिबा ना एडा आमर pyrimidine रे भी रहुचंती but adenine is the only case जोटा कि हमें purine रे देखिबा को पाउचे so purine base रे we can consider as adenine. See, so the purine रे guys कौन रहुची ना adenine and guanine रहुची but C here is not the option having guanine. Okay, so if the option is there guanine, then you can choose the answer as adenine and guanine or both A and B. Okay, so the purine base is considering about the adenine and guanine nitrogenous base of the DNA. Next, see, the law of segregation was proposed by Mendel after observing which generation? That means guys first amara kona law of assortment asthila then asthila law of segregation but law of segregation ta jo propose karithile but kichi gote study kariki research kariki end result re amara segregation result like law ta baharichi that kota guys that is the F2 generation. So what is F2 generation guys? This law also known as the Mendel's law of aggregation law of segregation first asthila then asthila law of assortment, right? Then independent assortment. Yes, the first law is the law of segregation. Sorry for the inconvenience. And guys, the each gene separates from each other, ensuring that each gamete carries only one allele for each trait. One allele for each trait. That means, come to the point that Mendel's law of segregation kaun kahuchi? Suppose there is a generation of two. That is the capital YY and the small yy. That is the capital is suppose the flower, you can have a look that the violet flower or the yellow flower. Okay, the gametes come to the that single capital Y and the small y, then they can form to have a capital Y and a small y. That is the violet and yellow. Suppose this is the color we are having the lavender. Okay, so lavender again they can form separately. That again se mane kaun paile na small y capital Y taku jete bele ame separate karuche ya fir joint karuche, the result we are having what? Either the same trait or the different trait. Either the same one or the different one, not exactly the same one, or the different one or the individual one. So individual different one pauche here is and individual again he jauchanti separate. Okay, this one is the F1 offspring from two kind of gametes. Then F2 re ame jetebele consider karuche kon pauche, suppose एठी हमें पर्पल को नेले एंड येलो को नेले ताकू जते बेले हमें क्रॉस सेक्शन करछे हियर वी हैव टू चूज दैट पर्पल एंड येलो देन पर्पल पर्पल मिसी की दीटा कोन पर्पल हे जाउचंदी देन अगेन यस येलो एंड पर्पल एंड येलो मिसी की दीटा कैपिटल वाई एंड स्मॉल वाई मिसी की सी भी पर्पल हे जाउचंदी देन जते बेले गोटे गाइस जे कोनोसी स्मॉल गोटे देखो रहउची लाइक कैपिटल वाई भी रहिबो बट स्मॉल वाई जदी गोटे रहउची सेठी हमर dominant character किए प्रेस करूची ना capital Y. That's why the color is suppressing about the capital Y. जते बेले double Y guys small रह जाउची that means the recessive is the small y. The different color, the yellow color is the recessive one whereas the violet is the dominant one. And जते बेले double Y मिसी की गोटी capital and small double दीटा जाको yellow मिसी जाउचंती गोटे different color show करूचंती. Here is the ratio is the 3:1 ratio. 3:1 ratio in law of segregation. After independent assortment रे हमर कोन आसीबो 3:1:1:3. Okay, so next देखिबा about what? Which of the following is a sex linked disorder? So what will be the right answer? Cystic fibrosis, sickle cell anemia, the hemophilia, the thalassemia. Which of the following is a sex linked disorder? Guys, the right answer will be hemophilia. See, sex linked disorder are genetic conditions linked to genes located in the sex chromosomes. That is about the X and Y chromosomes. We know the girls having the XX chromosome and the female having the XX chromosome and the male having the XY chromosome. So these disorders can be inherited in a dominant or recessive pattern. So, and they often affect male more frequently due to their single X chromosomes. Example, include hemophilia, red-green color, blindness, and Duchenne, the muscular dystrophy. So these are the symptoms we can use after the X-linked inheritance. So if the father have the XY chromosomes and the mother, as usual, you know, the XX chromosome, the transformation होउची gamete formation होउची no matter maybe the girl होबो केतेबेले जेतेबेले again XX वाला girl हे जिवो, that is the normal daughter having the 25% of character. Okay, the carrier character is 25%. Here is the linked recessive inheritance, but the mother is the carrier one, not the father, but here the mention is the mother is the carrier one. But there is a chance of what? 50-50%. So see, so again X सहित हमारा कौन normal X आसिला, so X सहित again Y आसिला, that means XY, the normal son आसिला, then XX आसिला, the carrier daughter, then XY, the after affected son. That means guys केमिति ହେଲା ना first the normal daughter आसुची, then normal son आसुची, then जो daughter आसुची that may be carrier or may be normal. Okay, but carrier हेबार chances 25% होची, but there is a full chance of a जो male affected, the full chance of affected having a affected son. Okay, due to the gamete formation. Next, see, in humans, sex is determined by, being just कहली guys, in humans sex is determined by the XX in males, XY in males, XY in females, or ZZ in males. Guys, first of all, the ZZ is nothing mentioned in the biology. So XY, guys, Y जोटा अच्छी है total female case रे ही absent अच्छी. Female have always XX in chromosomes. And guys, so XX in males, एटा भी हमारा minus अच्छी. Guys, males always have XY, female have XX. So odd one out रहिला कोटा XY in male case. See, so sex determination in human beings केमिति हमें करूचे. Suppose father रो कौन रहिला ना XY chromosome, mother रो रहिला XX chromosome. जेतेबेले हमर cross section होउची, the reproductive cell रो formation होउची, then in the case of zygote formation हमर कौन होउची ना when father have a X chromosome, from mother we are having X chromosome, then the girl child will be the female one or the child will be the girl. If you are having the X chromosome from the father, then X chromosome again from the mother, again it is the gamete formation of XX, the child will be the girl one again. Guys, if you are having the Y chromosomes from the mother, we are having X chromosome, the XY, the chances of male is also okay. And guys, when this is also X chromosomes and from the Y chromosomes, there is no matter like तमरो dependent किए रहउची ना father ऊपर की को chromosome आसीबो. If he is giving the Y chromosomes, then the result will be a male, and if he is giving a X chromosome, the result will be the female. As simple as this. Okay. Next, the structure of DNA was discovered by the structure like DNA तो हमें जानचे discover के करथिले, but structure of DNA के discover करथिले. Guys, structure of DNA was discovered by Watson and Crick. Discovered by Watson and Crick. See, so starting in the year, the DNA model structure guys discovered इथिला केबे देखो in the year 1953 रे by James Watson and Franz Crick created the 3D model of double helical DNA 1962. The Nobel Prize in Physiology or Medicine was awarded to the along with the invention. So starting ହେଇଥିଲା nuclein ରୁ, the nucleic acid ରୁ କେବେ 1889 ରୁ, then start ହେଇକି in the year 1881, 1990, 1950, 1952 and so on. The end ଆମର DNA stand ର structure है discover ହେଇଥିଲା. Okay. So next देखो, a change in the sequence of DNA is called. Guys, a change in the sequence of DNA is called what? Translation, translocation, mutation or duplication. Guys, a change in the sequence of DNA is called what? This is mutation. This is mutation. So what is mutation? See, so different types of mutation जेतेबेले हमर कोन जो नाइट्रोजनस बेस थाए से चेंज होंती डीएनए टू आरएनए को कन्वर्ट करबा पई. ओके, तो थायमिन कन्वर्ट हुए कोथिरे ना यूरासिल. So original copy of the gene sequence with TAC being the correct triplet code. हेजा जोटा कि wrongly format हुए ताको पुणी चेंज करंती TAC रे. That means हमर चेंज होची कोन ना AC तो रहुची T and A भितरे फर्स्टली कन्वर्सन होची. बट तो DNA टू RNA को होची सेते बेले कोन होची ना यूरासिल एंड थायमिन भितरे कन्वर्सन होची. ओके, so this is all about the process of insertion, then the deletion, everything you can have a look here. Okay, next see what is the full form of DNA, guys. Just ebe kahithile, I think this is the repeated one, the deoxyribonucleic acid. The human genome contains approximately जो हमारा base pair थाए approximately कितने base pair contain करे. The human genome contains approximately 3 billion base pairs. Approximately 3 billion base pairs. See, so what is, guys, human genome? The human genome refers to the complete set of genetic information encoded in the DNA of a human organism. It comprises approximately 3 billion base pairs of DNA, organized into 23 pairs of chromosomes, including 22 autosomes and 1 pair of sex chromosomes. That means in the 23 pairs of chromosomes we are having in our body, there is always a 1 pair of sex chromosomes. That if I am a female, I am having the X chromosome, and if you are a male, then you are having the XY chromosome. The X and Y pairing the 1 pair. Okay. So the human genome contains all the genes necessary for the development, functioning and reproduction of a human being. Understanding the human genome is crucial for unveiling genetic traits, diseases, evolution and personalized medicine. See, the Down syndrome is caused due to. The Down syndrome is caused due to. Give me a minute. Yeah, so Down syndrome is caused due to the trisomy of chromosome 21. Due to the trisomy of chromosome 21. See what? So causes of Down syndrome is what? First, the chromosomal abnormality asije. That means the trisomy of 21 as instead of what? 22. The mosaicism asije. The translocation show करे. That means हमारा body रा defectiveness देखा करे. Suppose हमारा nose रा place किसी different था कि side ही जाए था या फिर तारा proper growth ही न था. हमारा ear पाखे side टिके growth ही जाए. So this is all about the chromosomal displacement. So next, see, the unit of heredity is. The unit of heredity is the nucleus, chromosome, gene or allele. Guys, the nucleus, the unit of heredity is gene. The unit of heredity is what? This is gene. See next. Guys, what are the genes? A molecular unit of heredity, the name of stretches of DNA and RNA that code for a specific protein. This has a specific function in the organisms. That means inside our body, when genes are functions like a molecular unit of heredity, the name for stretches of DNA and RNA that code for a specific protein, that is called as a what? Gene. The cell first consists of a nucleus and the chromosomes. Then the chromosomes are already, guys, what? The DNA segments, and the DNA segments each set having a single gene. Then the genes are made up of what? Due to the protein synthesis, the process is occurred. So next, see, Which of the following techniques is used in DNA fingerprinting? Which of the following technique is used in DNA fingerprinting? Is that PCR? Is that gel electrophoresis? Is that RFLP or all of the above? So guys, in case of DNA fingerprinting, the PCR as well as the gel electrophoresis as well as the RFLP all are essential. So see what is the DNA fingerprinting is actually. So procedure that analyzes an individual's unique collection of DNA restrictions fragments. These are the DNA restriction fragments. So different peoples have the separate fingerprint we are using. So detected by the electrophoresis and nucleic acid probes. The common uses for DNA fingerprinting is the forensic science tool in criminal cases, the paternity cases, the determine origin of known materials, the botany and zoology studies. This is the use of what? DNA fingerprint. Next, see, the term genome refers to what? The genes on a single chromosome, total genetic material in a cell, the mitochondrial genes only, or the genetic materials of plant. What will be the right answer? The term genome refers to what? The total genetic material in a cell. In a single cell, the total genetic material is called as the genome. The state of gene is called as genome, we can say. See, what is genome, guys? A genome is the full set of genetic information that an organism carries in its DNA. Okay, so the study of any genome starts with the analysis of chromosomes. And guys, chromosomes are a bundle of DNA and protein found in the nuclei of eukaryotic cell. That is, the genomes are present in the eukaryotic cell. And guys, the study of any genome starts with the analysis of chromosomes. We can study over the genome studies. Next, see, the Tay-Sachs disease is caused due to defect in. Tell me the right answer in the comment section. The X chromosomes, the autosomal recessive gene, the dominant allele, or the Y-linked mutation. What will be the right answer? The Tay-Sachs disease is caused due to the defect in the autosomal recessive gene. So what is the Tay-Sachs disease? Right, for the father carrier of abnormal gene and autosomal chromosome, and if the mother carrier of abnormal gene and autosomal chromosome. The father is the like a black A and the blue A like I see. This here is also a black and blue. Like one is the healthy and one is the like defective one. Okay, so the offsprings are maybe the healthier one and the maybe half defective both in the cases, or the may be affected morely. One is the either there is a 25% chances of healthier or 25% chance of not healthy. There is a 50% chance of unaffected carrier offspring, but affected is 25%, unaffected is 25% is healthy, and unaffected chances of affected is 50%. Okay, so each pregnancy bears a 50% chance of unaffected carrier offspring, 25% in chance of affected offspring, and 25% chance of unaffected not carrier offspring. Okay. So see, what is the role of transfer RNA in protein synthesis. What is the role of transfer RNA in protein synthesis? That is, the carries genetic message, translates codons to amino acids, or the forms ribosome, or none. Guys, this is translates codons to amino acids. This is the role of transfer RNA. While I am discussing about the transfer RNA, I have already told you about the translation of the codons to the amino acids. So no need of the extra elaboration. Still, the transfer RNA or the tRNA is a type of RNA molecule that plays a crucial role in protein synthesis. Specifically during the process of translation, it acts as an adapter molecule, bridging in the gap between the genetic code in messenger RNA and the amino acids that make up a protein. That means there is a bridge, there is a connecting bridge among all. This is called as what? The anticodon atom, and the anticodons are using the transfer RNA. Clear? Next, see, which process converts messenger RNA sequence into a protein. Which process converts messenger RNA sequence into a protein? The transcription, the translation, the replication, or the splicing. Guys, this is translation. So the process convert messenger RNA sequence into protein, that is called as translation. See, so translation is a step in protein biosynthesis where the transcript messenger RNA is translated into amino acids. That means from the messenger RNA that is transmitted to have the amino acids. The steps are the initiation, the elongation, the termination for each. Okay. So next, what is a karyotype? Is a test to detect DNA fingerprint or the chromosome layout in a cell, the blood test or the RNA profile. Not RNA profile, not a blood test. So among the all, what can be the right answer? The chromosome layout in a cell is a karyotype. So see, what is a karyotype, guys? A karyotype is a picture of all the chromosomes in a cell organized and arranged by the size and the shape. So depending upon what the chromosomal cells and it's depending on the shape and size, it is the karyotype picture. It provides a visual representation of an individual's complete set of chromosomes showing their number, size and arrangement. So karyotypes are typically prepared from cells in the metaphase stage of cell division. Cell division na metaphase stage rahi ita karyotype cell division hai tha, where chromosomes are most condensed and visible. Metaphase stage rahi ko na chromosomal division hui and they can visibly clearly. So guys, this is all about today's class. Dekhabo again. Next class is on tomorrow about the class biodynamics. Thank you so much. Till then, take care of yourself and your parents first. Thank you so much. Have a good day, guys.